So here we are. And what we're up to this week is we continue to work on individual capstone journals. And as part of our discussion on Monday, we'll just check in with you on that as a class together and see how you're feeling about your topic and get a little idea of what each person is moving ahead on and how they're doing about that, because we are kind of all in this together, even though those are really independent projects. But then also on Monday, we're going to do another round of what we did this Monday, which is to pick an additional example of normal plasticity and attempt to dive into the cellular and molecular mechanisms. And we had some really good examples of that on Monday to guide us to finding more. And because what we see is that when we look inside the organisms, we see some important principles and concepts and mechanisms that are pretty common in this story of phenotypic plasticity. So I really appreciate the effort that you all did in selecting articles. And we're going to follow up on them. A couple of them didn't get covered on Monday, so I mentioned I'll talk about them a little bit today. And then try to find another example of that and try to make it different again than your capstone project and different than your example that you got this past Monday. Now, maybe you'll find another example that somebody else already covered something similar to. That's okay. We're just trying to accumulate more examples, and what do we want to do? We're using them both to populate our understanding of our course and what each of us is trying to do with the capstone project, and that is to identify something that has relevance up at that ecological and developmental biology level, and then actually pursue it into the primary research on the topic, and also the mechanisms, the internal mechanisms and the principles, and some of the common features, because that's what developmental biologists love to do, because they have such a wealth of understanding of the details of mechanisms that regulate normal development. So we did that on Monday. We'll do it again next Monday. And then just a reminder that we meet next week, and then following that week after that is a week of spring break. And when we get back from spring break, we'll meet on Monday, and we probably won't have a discussion. We'll probably have continuing topics, because that Wednesday we'll have our exam, and it's going to be an essay exam, just half a dozen questions to help guide you into writing about what we've been learning in class. And again, that has kind of a tension of having a sort of a double value for you, both learning in class about a broad set of topics in some detail, but also helping you get ideas and paths on your own capstone project. Okay, so that's where we're at. And so you'll see that Bryce.
You sent an email today that said the slides that basically the stuff that we talked about last class is going to be in our material, possibly on the exam. What another question based on what we did last time be on an exam?
So it wouldn't stand alone. It wouldn't be like, okay, Bryce told us about these spadefoot toads, so tell me all about them. Like that. It would be part of a larger topic. And a good example of that would be, Luke picked an example of an insect and phenotypic plasticity. And when we talked to him about the molecular mechanism, it's that ecdysone receptor. And so it fits in, that example fits in really well with chapter 1 topics, where there's several insect examples, and I'm actually going to talk about more insect examples today. And so that would be a good example that would fit in especially. The other thing, though, is that that you individual students can bring those individual topics that you learned some about to the exam. And so that I wouldn't necessarily force everybody to talk about, say, Ashton's topic, like that. Ashton could bring it up as an example, like that, So there's that kind of openness, and so, for example, there isn't a simple grading rubric for the exam. It's to try to see where you're at in, what you've learned, not so much in your capstone effort, but in these class meetings and readings, Yeah, so don't feel like you really have to dive all the way into other students' topic, but they're part of it, and you should be sort of assembling kind of an arsenal of knowledge. And so the questions will have some specificity to them. It'll be like, you got to talk about insects and the ecdyson receptor, but that leaves them still broad, like that. Yeah. Okay. Yeah, and other questions or anything that anybody's thinking about right now about where we're at? Okay, and so just a couple slides. Again, I posted the slides like I said I would on our Brightspace, and I mentioned that I should check a couple of times today that there was this odd thing where the recording stopped halfway in, and I think you were all here, and so didn't miss out anything. But that's the frustration, but just the reality of having this as a supplement and support for us. And then Bryce talked about this topic, and Emily talked about that topic. And each of them was quite distinct and good topics to bring up. Some of them in greater detail than others like that. That's absolutely fine. Don't feel like you have to find an example similar to one that we talked a lot about, because that's not what we're up to. We're just trying to find experience of diving into the literature and going after a topic and then being ready to bring that to class. So Ashton sent me a couple of slides before the class, and Juliet had some also, and we just didn't have time to talk about them. So I want to just talk a little bit about both of those, and I'll ask Ashton and Juliet if they want to chime in some about this. And so here's this first one. It's the one Ashton picked, and it's about a bird, a sparrow. And what it's about is about diet and the animals, when the little birds hatch in the nest, what they eat and how that affects their digestive system. And when I was reading about it, it reminded me something about like how in humans, when we're babies, we all have an enzyme in our digestive system called lactase that can digest lactose, because that's mother's milk, a sugar that's common in mother's milk and also in other animals' milk, like that. Then, for a lot of humans, that lactase gene activity and enzymatic activity present goes down in adulthood, because, as an adult, it was unusual historically, ancestrally, for us to be drinking milk and digesting it. And so that's a kind of change. But then in some humans, they've actually evolved the ongoing activity of that enzyme. And so we're going to talk about something similar to that. And the story is that they first described in this abstract that when a little sparrow hatches, for the first few days, the parents are, for the first few days, the parents are bringing food for them to eat. And typically, the parents bring some little insect-type food for the first 3 days, and then they start bringing, I guess, sort of like more like seeds. Is that right, Ashton?
Yes.
So they just have a natural shift in their plan of feeding these little baby birds. So in a controlled setting, the scientists fed these baby birds a diet that wasn't insect or seeds, but had a similar composition. One was a really high starch diet, so it would have more of a seed-like presence, okay? And they looked at the ability of the animals to develop. And the first message is, if they just had one or the other style diet, they seem to develop really similarly. So in other words, there was no phenotypic plasticity in most of their development. But the scientists did find a difference in the digestive system, and not a story of what the gut microbiome was doing, but what the animals were doing and the types of enzymes that were secreted into the intestine and the digestive system. And in particular, they looked at an enzyme called maltase. And so maltase is just a digestive enzyme that gets released in the intestine. Ashton says he thinks, it's just widely common digestive enzyme, like we have it, and so forth. But in this species, we see a phenotypic plasticity based on an environmental factor. What's the environmental factor? Type of diet. What's the phenotypic plasticity? a pretty simple one and a sensible one, a shift in the type of enzymes and readiness to digest those. Like that. And so it's an interesting example because, you know, we do want to see this range, right? So if you look back on Monday, Luke had this example where the aphids were going to either be winged or wingless, right? A big morphological change, a big developmental change. And that's true, but in this case, it's a small one, and it's a simple one, and a sensible one to be able to have that plasticity, right? And I would say that, well, and Ashton, do you want to add anything to that?
This group also did another study about the adult house sparrow, and I was very interested in the fact that they found the opposite results.
Oh, yeah.
They had plastic, what is it? Aminopeptidase N, but they didn't have plastic maltase, so it was flipped. I thought that was interesting.
Yeah, yeah. So how was the study different? Were they using the same species?
Yep.
And same diet options or different?
Similar diet,
yeah. Yeah, and do you have any feeling for why it would have been a different result?
Maybe because I have two theories. Maybe because the animal knows evolutionarily that it's going to shift from high protein to high carbohydrate, and it makes sense developmentally to have those switching options at that time. Or maybe that you can't have plastic aminopeptidase when you are a nestling because you need all the protein you can get because you're growing.
Yeah.
But I don't know.
Yeah, yeah. So a good point, though, is that a research group like this will find something and they'll really drill into it, you know, with some multiple publications to try to sort out, a really clear explanation for what's happening there. And it has broad relevance. So, for example, I started out with this story in humans about, lactose intolerance, because I think these studies are broadly relevant in the mechanism and the notion, that there could be switching of these enzymes depending on the environment. And then remember how Bryce brought up this example of evolution, where we could have related species, so 3 species of this spade-toed amphibian, and 2 of them had this plasticity. They lived in environments where that plasticity was valuable because there was variable amounts of water in the spring in their environments. And then the 3rd species had lost the plasticity and only developed in this sort of stressed out, rapid metamorphosis way. And so that's something, that these people might also be interested in running into, is to test some closely related species and see which of those have that phenotypic plasticity, and whether it is absent in some species or others, because it could be a hint of some evolutionary changes. Because when organisms undergo speciation, initially they're very closely related, and there'll be some feature that's distinct, and that feature is something that helps them fit into their particular ecological niche. So, Ashton did a good job summarizing this and really going through the bulleted story of the message of this. And so those slides are on the Brightspace now, and as is the link. And I think the links were all that, and that's really helpful, right? If you want to go look at them, right? Because you might say, oh gosh, this is actually relevant, to your capstone project. So you might pick up one of these examples, even though it came up from another student or from class, and pick it up, use it or use it as a way to get further into, the tops of your capstone project like that. Okay, so great, and just a really good summary and a link to that primary research article. So again, not required to do it just this way or that way for next Monday or for your capstone. But try to dive into the mechanism some in one of these examples. Great. And so some data slides showing that, and like I said, Ashton summarized those. So then Juliet also has some slides and a different article. And oh, I meant to try to find this. So this article is about that seasonal change in fur color, like that we show the book has, where it's like, oh, the weasels have brown fur in the summer months, and then they molt to a different fur color, white, in the winter to blend in with snowy things like that. And so this is diving into that topic, I would say, into those mammals and looking at the pathway for regulating that. And it starts out with day length. So that's what they're measuring, and that's a reliable cue, and I've mentioned that before. Some people call that a trivial cue because it's not really what they're adapting to. They're not adapting to the day length. They're using this trivial cue to adapt to a changing environment that's predicted consistently over evolutionary time by day length. And lots of species do that, so they don't wait around till it snows or wait around till it's cold. They use the day length as a cue, an indirect trivial cue, to make a change, to make a switch. And that's a predicted adaptive response, right? Where they're like, okay, it's gonna get cold. And so really nice examples here of some nervous system and hormonal pathways that we haven't talked about that are relevant for this. So the pineal gland is a really important regulator of circadian rhythms, which is what we're talking about here, right? Biorhythms or circadian rhythms are set by short day, long day. And then you might have heard of this melatonin, because some people take melatonin supplements to help them sleep, like that. So a regulator of that circadian activity. And of course, these are vertebrate mammals, and so we have all these as well, like that. And so a good summary of those pathways, to introduce that is another example of how the nervous system can monitor the environment and then communicate that into the animal. And lots of times it's really important for it to be what's called a systemic response, or a response that would be governed by a hormone. And a good example of that is those beetles that were either going to have a horn and have one set of behavior based on a brain that wants to fight and dig holes and roll dung balls, or a different body, a hornless body, and a male that wants to dig a hole but not roll a dung ball, not fight, like that. And so you have to have really good coordination between the development of different organs, different tissues in the nervous system, like that, and the whole organism, okay? So really good example of that. And then I'll ask Juliet to explain this a little bit, but what they're looking at is that and is it kind of represented here? So this, I think this is a singular one too. So this paragraph over here, it says temperature was also shown to affect speed of weasel motes, but it's still mainly controlled by photoperiodic signal. So that's the idea of, this is going to be an issue in a changing environment. So we're having climate change, warmer winters, less snow color. That leaves these animals very vulnerable. Do they have any backup mechanisms to try to adjust to it? And I guess temperature is potentially a secondary environmental factor. And temperature is an environmental factor that we've talked about a little bit. We've talked a little bit about temperature and sex determination in some reptiles like crocodiles and turtles and fish species. But here's another example where temperature can also be a regulatory role. So good summary here with the slide shared. Juliet, do you have anything else you'd want to comment about this?
There's one more slide which gets into the changing environment, where they found the MC1R gene is pigmentation, so that's you see melanin production, which is a dark pigment, and then the antagonist ASIP is for pheomelanin, which is for the lighter pigment. And this is a separate study that basically found, through a study with a bunch of weasels that they had in museums,
Mhm.
And they like analyzed all their DNA, and they basically found that there was a subsection of weasels that kept their brown fur all the way through, and it was basically in the MC1R gene. It's a mutation in the amino acid sequence that switches, I think, like a lysine to a leucine or something, and that keeps them brown. And it was shown that like the weasels in those populations were in an environment that wasn't getting as much snow as it used to, so they think that it was like a selection for better like adaptation to their environment with a warmer temperature, where the white fur wouldn't have been as advantageous for them.
Yeah, and that, so that ends up sounding a lot like Bryce's topic, right? Where we have some, in Bryce's case, some closely related species. In this case, is it really the same weasel species, but in some different, in some different regions, and in a region where there was very little snow color, lots of the weasel had a mutation that caused them to just stay brown year round. So that's a really good, excellent example also, right? Where we see that kind of role in potential evolutionary change. I think I added this arrow. Oh, so this I wasn't familiar with this word, museomics, right? And it's rooted in a kind of a common term that's used now, where when somebody is studying DNA and they're going to study the whole genome, which is done very often now, that's called genomics, and that was the original omics term, genomics, great. And then some people are like, we should just be studying all of the genes' expression, so all the genes that are being transcribed into RNA at this moment. And so that's studying the patterns of gene expression, and people cutely call that transcriptomics, because we're going to study the transcriptome, which is all the genes that are re-expressed at any moment. And then the next famous one is, wow, it's really also interesting to look at the situation for all the proteins, because it's not just that the proteins are there, but some proteins are activated by phosphorylation or inactivated or other modifications. And so scientists are trying to study the status of all the proteins. And in that case, challenging, because it's not just are the proteins present, but what's their activity status, like that. And that's called proteomics. And so this museomic is kind of a cute use of that. And what it comes from is this, which is a very interesting thing that Juliet mentioned, and that is it's using museum specimens to start to be able to collect data from history. Because there are some museums, right, that have had like a little weasel in a display case for 80 or 100 years, like that, you know? And they actually went to those samples and got data from those samples. And so they're emphasizing that when a person wants to do what's called a longitudinal study, right, how can you do that? You know, we haven't been collecting data on these little weasels for 100 years. Is there any evidence anywhere of the, you know, history of these weasels? And the answer is yeah, in some museums. And and that's actually a pretty common thing, not limited to this topic, but for example, I know that some museums have collected a variety of shells, like seashells, either freshwater or saltwater seashells, over a really long time. And some ecologists are going into those, and they're able to look at the mineral composition of those shells and actually understand what was going on in the ecosystem where they were collected, right? Because a lot of times there'll be a museum that is associated with a certain watershed or something like that lake, things like that. And so just an interesting thing that doesn't have immediate relevance for our course, but just is, you know, broadly interesting like that. And so it says down here at the end of this abstract, it says using biological collections as valuable resources to study natural adaptations, our study showed a new evolutionary route generating winter color variation in mammals, and that season camouflage can be modulated by changes at single key genes. So that's, I think, where they found that idea that, oh, here's some guys that stayed brown all winter, what's the deal? And they're like, well, we have some old specimens in a museum, we can bring those out and analyze those, that data and those too. And probably looking at the protein sequence in the actual, fur, individual fur or hair fibers, like that. So, that's not so immediately relevant for our course, but it's a really good example. So great. And anybody else have anything else they want to add or bring up from carrying on from our discussion from Monday? And so we'll try that again this coming Monday, where everybody will try to investigate a little bit, something different than, certainly different than a capstone project, but also, see if you can find something that's a little different. Again, if you find out, oh, it's another bird, digestive story, but you find it's, wow, this is another good one, we should talk about it. Just go for it, it's not to be like we're going to get shot down because you're like, oh, that's just what Juliet talked about animal fur also. There's lots of different examples, and it actually would be nice to have some that are overlapping because that helps you bring more things together for a topic. Okay, so I just want to mention this because I mentioned it and just show you a little bit about this briefly. We talked about this idea of a transgenerational effect like that, and scientists are very interested in that. So a transgenerational effect on phenotypic plasticity. In the case of the aphids, the female's offspring were what were especially affected that we looked into. And so how can you have transgenerational effects? What are the molecular mechanisms? And by far the most common one that people are interested in is it possible that patterns of DNA methylation could be inherited from one generation to the next? And they can. And so when people are looking at these transgenerational effects, they often look at the patterns of DNA methylation. So I just wanted to show you this, and it fits into our course, but it's a human story, so it's the data is limited, right? Because they can't set up experiments on humans, but they can take kind of like museum omics. They can take history and evidence from history of what's happened and start analyzing what happened to these humans that experience these different environments. So I just want to show you that. And so the idea is transgenerational changes in DNA methylation patterns in humans. And it starts out with an example. And the example would be something happened to a person's grandparents or great-great-grandparents, and it seems to still be impacting the development of their offspring through a few generations, okay? So pretty interesting stories that fit into our course. So just to look at some of these that I just picked up, from sort of AI overviews. So transgenerational changes in human DNA methylation patterns are primarily documented through these epidemiological studies of cohorts exposed to severe environmental stressors such as famine and trauma. And I guess I got that covered up in my screenshot, but these changes involve chemical modifications of DNA specifically added to the groups of CPG, so methylation, DNA methylation, okay? And then just to show you some examples, and this one is in our book, and it's in a later chapter. Our book actually uses it as one of those things where it's in that second half of our course where the disruption, but I think it's misplaced there. I think this is a natural feature of humans where we have this flexibility, and it's referred to as the thrifty phenotype. And it's this example that I described briefly in our course before, and it's true of mammals. And that is the offspring, the fetus, embryo, and fetus are being carried in the mother's womb, and that mother's environment can affect their offspring. Okay? And so that's one example. And it happened during World War II. That was a long time ago. It happened to a really specific set population of people. During World War II, it was the country of Holland or the Netherlands or the Dutch people. That's all synonymous. And it affected the fetuses that were growing during that period of intense starvation that occurred over about a 10-month period near the end of World War II, where the Nazis were in charge, occupying the Netherlands, and they just starved everybody because they got tired of the resistance that was happening in the country, like that, okay? So that's an example. It's in our book. And then there's some other famous examples. So Holocaust survivors. Another example of a long time ago in history, a set of people who lived in an environment that was a good environment prior to the war and a good environment after the war. That means good nutrition, good medical records, good, healthcare and medical records. And so you can look back on those epidemiologically, and you can follow changes over these, in these populations of people in specific gene expression patterns and associated DNA methylation. And so then here's another one in China, where historical famines continuing to impact offspring like that. And then here's the last one, and it's, I think, in northern Scandinavia, Sweden. So there's a thing where they live up near the, closer to the North Pole, and so their growing season for crops is really limited, and it varies. And so there's a documented history over a very long time of times when they suffered from real famine, and then they look at their offspring over a few generations like that. Yeah, and so just to show you a little bit about that, because the specific examples in the aphid, wingless aphid is great. Other specific examples are great of transgenerational effects. But some of this research is driven forward because it hopes to help us understand how this affects human health and disease, like that. So just a little bit about that, and because some students are really would be interested in this. Some students are particularly interested in biomedical examples that would fit into our course, and there are some really good ones like that. Okay, so great. All right. So then I also wanted to do this. So the rest of the time today I'm going to talk about another insect example, and it's just going to be talking about an insect example, and it's going to be focused on this ecdysone story. And so we saw in Luce's example that it is part of the story, but nice flow chart, right? Nice mind map, concept map showing, we have some nice different pieces of it coming up. And ecdysone fits into that, and our book has ecdysone fitting into the dung beetle story and the butterflies with different wing colors and other examples of that. And it turns out that I study insects, and so I want to talk about an example of this in the research that I have done, okay? And so it goes like this, where we're talking about that nuclear receptor gene family, not in vertebrates, but in an invertebrate, in all arthropods. All arthropods have a hormone that's called ecdysteroid or ecdysone, and that's fairly synonymous, and there's a receptor for it that fits in. So it's very relevant for those organisms, but it also has broad relevance in understanding that nuclear receptor family in animals, like that, okay? All right, and so here's just that image from the book, from chapter 2, where the book was, diving into molecular mechanism and saying, yep, there's a hormone, it's going to get into the cell, it's going to bind this receptor, and that receptor is a transcription factor, so you can watch that hormone's action by changing gene expression. The genes are getting turned on and off like that, okay? All right, and so the book dove into some of these molecular mechanisms of gene expression, and when we talk about genomics and transcriptomics and proteomics, we're talking about being able to study that story in a big scale, big data way, okay? And so we saw that in the adult males, and we could see how, and this is a really good thing to be aware of, is that once we know a pathway of molecular and cellular signaling, like a hormone, well, we can manipulate it in an experiment. So these people just gave injections to those males as they were developing, and they could change the phenotype of the males. And I mentioned before that we have this idea that phenotypic plasticity leads either to a polyphanism, that's what this is, one or the other, discrete differences between populations, or reaction norm, a continuum of change. And these people could use those regulation of the duration and level of ecdysteroid to make that polyphenism, discrete changes, into a reaction norm. They could just get males all the way across the continuum. And that was also true of the butterflies and so forth, because they're manipulating the internal pathways in the story, okay? Like that. And so, their book talks about that. We have the table in our book about a variety of examples. Luke brought up the aphids. There are examples of them fitting into there too. And there's evidence in the wing versus wingless form that ecdysodes is playing a role, and it's levels. Okay, all right. So, I'll put it over here because. So now we're going to talk about this species I study, and I hope I forgot. I may have forgotten to put in a figure. Yeah. So this is going to be a story about a moth. And the moth lives down in the Carolinas and Virginia, and it's called Manduca sexta. People just call it Manduca. We have a colony of those here at USM. I brought some over with me. We'll look at them, right? Because we have them growing here, growing continuously. And I study how hormones regulate insect metamorphosis. How did I get going on that? So a long time ago, in college, I ran into the topics of molecular biology, and I just instantly fell in love with them. And molecular biology is merging of biochemistry and genetics, and it occurred in the 1940s and 50s when scientists discovered that DNA was the primary heritable molecule, and they discovered the structure of DNA. And that brought together genetics of inheritance with biochemistry of biochemical molecules like DNA. And so that's molecular biology. And I'm a molecular biologist because I fell in love with that in college, just in books. I just picked up a book in the library and I was like, oh my gosh, this is the coolest thing ever. And it's molecular mechanisms. And a molecular biologist, I mentioned before, because Ashton agreed with this or was the first to make it, they don't really care about the organism. Like, oh, the sparrows eat different kinds of food. They're just really into the molecular mechanism. So it's like a person, like a car mechanic, who's just really into the details of car parts, and they don't care that you're going to go to Florida, on spring break in your car. They're like, whatever. The car is cool. The carburetor is wicked cool. How does that work? Like that. How is it different? So that's what I'm interested in. And so I went off to graduate school and learned about molecular biology. And the lab I was in, we studied transcription and regulation of transcription, and we didn't care about what it was in. And I happened to be studying fruit flies, but most of the times it was just fruit fly cells in a dish, and it wasn't even the animals like that. And I mentioned that because we thought, my lab thought those people that studied the animals were just dorky losers like that, And but then I finished grad school, and the whole time I was like, I hope I could be a professor. I really liked the professors where I went to college. I was like, that seems like a cool career. I see these professors talking to each other, and like learning all about stuff their whole life. And so to do that, a person typically gets trained in grad school, gets a PhD, and then they go off and do something called a postdoctoral fellowship. And the purpose of the postdoctoral fellowship, at least in the context that I was trained, is to develop some projects that I could take with me to a job as a professor, right? Because the professors are like, what are you working on? What can you get grants to study? Like that. And so I ended up going from New York to Seattle, where there were a couple of big labs, a husband and wife team, that were studying Manduca sexta. And those people were not molecular biologists, and I showed up there being a molecular biologist. And so I had this attitude, like I got into this lab, and I'm going to go do something there. But I thought most of the projects and the way they talked about stuff were not interesting, because I was intentionally diving into that setting. And I'm kind of pointing toward Ashton, because Ashton brought this up, like I don't really care about, what's going on at that level of the organ or the organism or the environment like that. I had that attitude too. I'm just like, I just want to move on the nuts and bolts. And my attitude was, it turns out this Manduca is a typical insect that goes through a typical insect metamorphosis, and that makes it's very different than a fruit fly. A fruit fly goes through metamorphosis really fast. And so everybody knew a lot about fruit flies, and I would take that knowledge and I was going to use it in Manduka and find out more about metamorphosis in Manduka, and then probably go back to fruit flies, right? So I started studying it, and it's good to point out because that short, just comment that Ashton had is a big part of the story of my life. Because I kind of fumbled around in that lab for a couple of years trying to figure out what I should study, okay? And I was doing different things, but it wasn't really good big projects to be like, yeah, you found some stuff out and I bet you can get a grant and go off and get a job as a professor and study this stuff, And that was my big goal, And I understood that, but, I didn't have people there to talk to that were really focused on the same angle, that I was trying to bring like that. And it was a very good lab, lots of people, because it was multiple labs, and they were raising these animals. And so one thing that was happening there that was just, to me, a kind of weird curiosity is they're raising them in lab like I am, and the rooms are kind of like big closets. There are no windows in there. The temperature and humidity is controlled, and the timing of the lights. And in Seattle, they had a big production, and they had 2 of these rooms. And in one room, the lights, the timer they were on for 17 hours a day, and the other room, the lights were on for 7 hours a day. And they had both those rooms going. They called one room the long day room, and everybody did their experiments on long day room animals, including me. And then there was this other short day room. And I knew why it was there. It was there because they do this overwintering thing that's based on day length. And so, let's see if I have the word here. Yeah. So it's a kind of hibernation, and a lot of species do this, and it's called diapause. And diapause is a type of hibernation that includes a developmental arrest. So at the time when I was looking at working there in Seattle, it's a long time ago, right? I've been a professor here for a long time, and there's no way anybody was talking about ecological developmental biology as a field. There would be people that would be like, yeah, these guys overwinter. They live in climates where they can't be active during the winter. All the insects here, except monarch butterflies, have to overwinter. They don't move away. They're overwintering right now. And there's a closely related species, another Manduka genus species that lives here. And it's overwintering right now as the pupa underground. And they're just waiting like that. And it went into diapause because of day length. So it made a predictive adaptive response. So that's a perfect topic for our course, right? But there was no such topic then. And I just thought it was weird, right? Like that, okay? So then I gotta just show you, let's see, what else? So then I'll just show you this. It's just a detail, but this ability to overwinter appears to have evolved independently a lot of times. So this just shows some closely related species, and it shows that whether they diapause or not. And they've evolved diapause, and you can see they've evolved diapause at different stages. And you see that around here. So if you went out in nature around here right now, you would find some insect species that are present as eggs, really embryos, like in a leaf or somewhere out in the environment. That's where that species is over winter, every winter. And what happened there is a transgenerational diapause. The mom was detected at day length, and the eggs that were laid included embryos that arrested their development. And they're just paused like that. And then they'll resume. And they're not paused because it's cold. So remember, in Seattle, they had the 2 rooms, and the animals would go into diapause in the short day room, even though it was just normal room temperature, because it's just day length is the cue. So they would just go and they'd arrest, right, like that. So a really curious thing. And then there are some species that have evolved a larval diapause. So you could find like a little curled up caterpillar or one like curled up inside a leaf or something like that, or maybe inside some bark on a tree. And then some of his adults. And what this doesn't have is a really common one, and that is pupal diapause. That's where my species, Manduca sexta, undergoes diapause. So the animals in summer or fall go do 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 do, and the caterpillars are monitoring the day length, and if the day lengths are short, they form a pupa and they just stop. In my species, the pupa is underground, and there are lots of moths that do that. So you know there are some species of moths that spin a cocoon, they're hiding. It's just a blanket like that. And you can find some of those cocoons around, and there's a pupa in them, and it's just hiding. And my species doesn't spin a cocoon, it digs a hole under the ground like that, okay? So it's the pupa that diapauses. And if you open up one of these pupae early in diapause, it just looks like soup, because the caterpillar gets almost completely destroyed by programmed cell death. It's not a soup, it's more of a stew, because some of the organs are maintained in this stew and are going to be a continuation from caterpillar to adult. For example, the brain. The brain is floating around in there in an otherwise just soupy setting. And really curiously, that's when this animal diapauses. So it's a developmental arrest, and it arrests during a time when there's tremendous changes happening. The adult body parts are growing and getting organized, and the caterpillar parts are just getting destroyed, and at a certain moment it just stops, right? And I didn't know that at the time. I just knew there was the room, and there was, those animals were going to shut down as pupae, right, like that. So I'm working in Seattle, and this guy, a scientist, a professor in Ohio, publishes an article, and he says that, and it just was a very simple article, not in a big journal, because he just made a very simple point, and he says the reason that this species diapauses is because it stops making ecdysone, so the hormone we're learning about, this ecdysone or ecdysteroid hormone, and it stops making it. And I read that article, and it was just an article about Manduca sexta. There aren't that many of them. I'm going to read them all, kind of like that, And I read it, and his first name was Dave. I thought, oh, that's good. I like that, But other than that, it was just this thing, But then I started thinking about it, and I was like, that is just weird that they stop if they don't have ecdysone, that they just stop. And so we had purified store-bought ecdysone, and I just went in the short day room one day and I just gave some injections like they did with the beetles, but this was before that work was done, and I wouldn't have seen it anyway, but I was just like, and the animals resumed their development and came out as moths, like that. You could interrupt this diapause just by giving an injection, and it kind of got them going again, like that. And so I was like curious about that. And so then again, we had this purified hormone. And so what I did then is I just open them up and I'd find the brain and I'd open them up in a sterile condition and I'd put the brain in a dish with nutrients and keep it sterile. And then I'd just add hormone or not, different levels of hormone. And I did that, and I could see that up above a low threshold, the brain would start growing again, because I could measure the cell cycle and the proliferation of the brain, and I could just turn it on and off repeatedly in a dish. Because insects don't have blood vessels, and they don't breathe through through any lungs, so they actually survive really well if you just take a piece of them, as long as it's sterile. If you just put it in liquid nutrients, they'll live for months, and they'll grow like that, yeah. And so over time, for example, I learned how to grow a great big moth eye in a dish just by letting it go and giving it enough hormone. But that would happen, and there was this threshold. And if it was above that threshold, they would grow, and if it below it, they'd stop. They'd just go on and off. And I studied a couple of different kinds of tissues. I studied the brain. I also studied the eyes a lot as the eyes grew. And that was the short day room. And the long day room animals, they never stop. They're not going into diapause at all. They think it's summer, the days are long, and they're just hurrying it up. Gonna develop into a moth, reproduce, and go through another cycle of, another life cycle as quickly as we can. But when I took those tissues out of the Lundy animals, they had the exact same threshold. They were also requiring this low level of the hormone to keep growing. And if you dropped it below the threshold, they'd stop. So that was in the dish, very consistently with Dave from Ohio saying, yeah, if they stop making ecdysone, they'll stop. And so I'm just like, what is going on with this? And the reason I was saying what is going on is because this ecdysone has been known for a long time, because the word ecdysis is the word for molting in arthropods. And ecdysone is the molting hormone in all arthropods. All the insects, all the crabs, the lobsters, the ticks, the spiders, they all use ecdysone. You can call it molting hormone, that's what it is. And that's weird, because when the animals molt as they grow, they make a huge amount of ecdysone to molt, and it triggers the molt. And so I'll just show you an image of that. So this just shows that in fruit flies. The little larva is growing and molting, and you see it has these peaks of ecdysteroid. And that's what people had studied for, 50 years, like that, That's why they found the hormone, because they're like, dang, it's molting hormone, there it is. And here I was using it, and the animals were not molting at all. And they weren't responding to a big peak. I have, the other people had studied the level of the ecdysone in the hemolymph, the blood of Manduka, and they showed it had to be a really high threshold to cause a molt. And I was down here at this very low threshold. And in a molt, it just takes a exposure for like 2 hours of a high dose, and the animal will molt. And in endocrinology, endocrinologists call that molting response a ballistic response. And that's like where you shoot a gun and the effect is going to just keep going, right? They're going to have, somebody's going to get hurt, and they're going to go to the hospital, they're going to be continuing things. So they call that type of hormonal response a ballistic response, because it just requires a short exposure to the hormone, and you can have extended responses. The hormone does not need to stick around. The other kind of response is some hormones require continuous presence. And that endocrinology term for that is, it's called a tonic response, an ongoing requirement. And that's what I was working on. Diapause involves not a ballistic molting response, but an ongoing lower level of hormone is required for this tonic response. And you can see here, and I picked this one because it shows the peak, but it shows you that the hormone never goes down to 0. And these levels down here are enough to cause the growth, the ongoing tonic requirement for metamorphic growth. And so I published a bunch of articles about that in Seattle, and that became one of the projects I could bring to USM, like that. And I had 3 different projects, and came here writing grants and start working on them. And it turns out that another one, a project that I got more progress on at USM. So I haven't studied this one for almost since I got here. But it's fascinating to see, because this whole time, I'm still, even to this day, I'm still like a friend with Ashton, and the attitude of like, I'm just interested in this molecular mechanism. Because for me, what's really interesting is these cells, these are target cells that are responding to the hormone, and they make different responses depending on the concentration. For example, in the brain, what's happening is a moth and a caterpillar have the same brain, but a moth brain is much bigger. So during metamorphosis, the brain is growing a lot, and there are stem cells in the brain that are dividing a lot. And when I say they need hormone to keep dividing, their cell cycle requires the hormone. And if you take the hormone away, they stop in the cell cycle. And for students that know about the cell cycle, it almost always stops at this point in G1 called start. These stop in G2. They need the hormone to go through G2. So another curious publication that these guys are regulating a very different point in the cell cycle than most animal cell cycle stuff. And that would be their diapausing. They're just stopped. And then you add the threshold back and they go. And then one day in lab, in tissue culture, brain culture in lab, I just accidentally had a new dilution of the hormone and I added it and the cells didn't grow. And I looked back in my notebook and I discovered I had made a miscalculation in dilution and I had given them a big dose. And so I discovered that second threshold is relevant for these cells because the second threshold causes them to go through programmed cell death. And so there's a period during metamorphosis where the animal is going through neurogenesis, new neuron production, new brain growth, and then it's going to be done with that. And the stem cells that do that require tonic, low levels of ecdysone continuously to go forward. You can evolve pretty easily diapause by just taking away the hormone. Right? Remember, the hormone evolved independently a bunch of times. It must be easy to evolve. Oh yeah, that's what this hormone's doing. At these low basal tonic levels, it's driving everything about metamorphosis forward. Because I've looked at other tissues, and remember I said I looked at the eye. The eye is doing a tremendous variety of developmental steps, making the eye. The eye is one of these multifaceted compound eyes like a dragonfly. It has like 10,000 little mini eyes. And the little mini eyes are being made in a wave. And the wave just stops. Like if you went to the ocean and you saw, oh, the waves are frozen like that. Just a freaky, crazy thing. But that's what hormones can do. And this is a long time ago. And I didn't, I'm not sophisticated enough to be reading all this literature. But it set the stage for all of these stories. Their stories are facts, but the explanation for why is ecdysone involved in this make a wing or not make a wing, or a horn or not a horn, it's because of this tonic role, right? That you have to have it, and the levels matter, and the duration matters like that. So then in the neurogenesis, these stem cells, when they're done making the brain, they actually all respond to a peak in the pupa of the hormone, and they go through programmed cell death. And in the dish, I could expose those stem cells to either level of hormone at any time. And those cells were ready to respond and either grow and divide or go through programmed cell death to the same hormone, same cell thing. And that's what I'm really interested in to this day. Like I said, I got going on another huge project, but I would like to go back and figure out what's going on? They must have a different form of the receptor. One has a high affinity, one has a lower affinity, something like that. What is the mechanism there? Because it's broadly significant like that, And it sets it up. And it's an example for our course, And before I taught this course, I was studying that stuff for a long time. And when I started teaching this course, it still didn't strike me that I was studying this topic, like that. Because it's just, a lot of times, you're going down your path and you just don't appreciate, oh my God, no, I'm like one of the early people who studied this freaking topic in insects, right? Because it is developmental plasticity. Either you diapause or not. And the switch is just whether you stop making this ecdysone or not. And it's a funny thing because we're talking about molting hormone. And the idea is that molting hormone in all arthropods, that molting hormone evolved a long time ago, additional mechanisms for regulating arthropod life cycles, like that, right? Okay, like that. Okay, so let me show you a couple more things. And so one of them is, I'll show you. So here's the idea. So we're doing great, and this is just, it turns out it's just very relevant for this course. So here's an example of those compound eyes, multifaceted compound eyes. They're in a housefly, dragonfly, they're in this moth, they're in, lots and lots of insects. They're in a fruit fly. And people have learned a lot about them in the fruit fly. In the field of developmental biology, fruit flies are very, very famous fruit fly geneticists named many, many genes that are found in us because they're also found in fruit flies. And one of the most successful strategies to genetically analyze and identify developmental regulatory genes like hedgehog, which became sonic hedgehog, and Wnt, and so forth, was in these eyes, because in the pupa, when the brain is growing, the eye is growing. And the way the eye grows is it first grows rapidly to make a big sheet of cells that are just all uniform and are committed to be an eye, but not a specific cell type. And then, starting at one edge, a wave, like a wave on the ocean, of development sweeps across the eye, and in front of it, it's just a plain sheet of cells, and behind it, you see all these little mini eye units. And each eye unit is made up of 21 or 23 cells, and they include cells that make a lens for focusing, light, they make photoreceptor cells, neurons. They also make some screening pigments, to make each one a little kind of like a cone of eye. And it just makes lots of them. And that's what my animal does too. And that wave is crossing in the pupa stew, crossing the developing eye, and it just stops during diapause. Again, in the short day room. It's not cold, it's just that the hormone's not there. So I could start it and stop it again and again in a dish. And it's not just the cell cycle, it is many, many different developmental stories. And when you zoom in and you look at the cells under a microscope, they really look like everything just stopped like that. So can a hormone do stuff like, coordinating all these disparate parts of the stories like that? We gotta have the beetle have the hormone affect both its brain development and its horn, like that? Just amazing to see what a hormone can do. So this is another example, or a good example in our course of, sure, hormones are involved because hormones are just grand regulators of just huge chunks of development. So for example, when an animal goes into diapause and comes back out of it, like that, there's no scar on the eye from where, that development arrested. There's no program cell death that has to clean up that. It just stops, and it goes. And like I said, I get to start it and stop it repeatedly, and many, many times in a dish like that. So super fascinating. And another thing I found was this thing where, and these are just different parts that I studied, like that, and the brain as well, like that, to look at different ways the adult is growing, and it's all growing, they're all growing the same way. So I'll just show you this too. So you've heard me say over and over again, because these articles talk about it, they'll call the hormone ecdysone or ecdysteroid. And it turns out that's because there's a funny naming problem with the hormone, and it's a historical problem that continues to this day. And what it's like is, probably in the 1940s, they were like, we're going to try to purify this molting hormone, and it's this ecdysis molting process in arthropod. And so they did it, purified it in insects, and they purified it, and they said, we're going to call it ecdysone. So, it's a hormone that causes ecdysone, we're going to call it ecdysone. And so there's a molecule they purified, and we now call that molecule E. It's still called ecdysone, and it's a molecule. And just like a year after they had purified that, they're like, oh, actually, like with a lot of hormones, it's a family of molecules. So a lot of times hormones will get modified, like we talked about thyroid hormone, it gets modified, and there are some active forms of it, some inactive forms. And like in less than a year, again, 80 years ago, they discovered there's another form, it's called 20-hydroxyecdysone, and it's just this E with hydroxyl group added. And then they found in all different kinds of arthropod settings, the E is an inactive precursor, and the 20E is the active form of the hormone, like that, And I found, great, I was using 20E. But remember I said, I could kill the cells through programmed cell death. I would give them a big dose of 20E. And 20E is the real active molting hormone. And you can see here that I got the, measured levels of these because originally the the peak, right? It looked like this, right? Oh, there's a big peak here, you know, we're gonna have a molt. And then in the pupa, there's a gigantic peak, and it was just a big, long, black line. But I found that, right, the brains would divide during this time. They'd be dividing along, and then they'd stop. And in a dish, they'd stop because they died to program cell death, but I think it goes at 20E. So it's like, wait a second, there's lots of hormone right here. Why aren't they dying earlier? And so, you know, measure the titer, measure the levels of these hormones, and I found, oh, it's because this is the precursor. And the real peak of this active multi-hormone happens later. And it fits the timing in the animal just perfectly, like that. Like right here, yeah, those brain cells die, they're done, but also the eye has finished making the pattern, and then it goes through this type of differentiation. The cells have already differentiated, but they kind of go through this next type of differentiation that is a kind of tissue maturation. So for example, the neurons have already started forming axons going back into the brain, but this is the hormone that causes the lens to form and causes the pigments to form and causes the neurons to start making the microvilli that are going to capture the light. So it's a kind of tissue maturation thing. So it's just a bunch of cool biology. And I found that, you know, and I was like, oh, this is in inactive form, cool, that all fits together. But then I was like, I should probably test this E, the inactive precursor. And what I found is in a dish, only 20E could drive the molting, like the high dose like responses, the programmed cell death or the development of the maturation of the eye. But either E or 20E could stimulate the cell cycle. And so really fascinating, because just another level of regulation where for these other tonic responses, these basal responses, these ongoing responses, it just could be the precursor form, and the precursor actually active in those settings like that. So just a whole bunch of biology comes tumbling out from this. And again, I wasn't driven forward by, oh, I'm an ecological developmental biologist, you know, at all. I was a molecular biologist who was becoming a developmental biologist, but still not thinking about really the environment. But that's why, you know, when Action brought that up, I was like, well, you know, in my life, I have that same attitude. But I also kind of got to cut this sort of tough love from my experimental results, where I'm like, poosh, the environment is a really big deal, you know, like that. You know, I would go in the building and there would be the long day room and the short day room, and I'd be like, I don't know why we have a short day room. I think it's just because they had an employee taking care of the bugs, and they're like, we gotta keep him busy, you know, like that. Because nobody was using them, you know, but except then I started using them. I'm like, God, this is a fantastic resource, like that. So anyway, I think we got to stop in a couple minutes. But I just want to show you that, and I'll show you also, just as a little follow-up on this, is that eye development, just to let you know. So the caterpillar has some little tiny eye units. They're called stomata. They're just like half a dozen little eye units like that. And the adult eye grows from cells that are right next to it. So there are two separate organs, a larval specific, caterpillar specific eye, and then this great big adult eye. And in the pupa, that's the pupa, and that's the size of the adult eye, and that's when the pupa is going to be like sort of still almost a stew. But at this time, when it molts, you can actually see a mark where that line is of the wave. So the wave goes from the posterior to the anterior. And when it pupated, that's right where the eye wave was at the front edge of it, like that crest of the wave, like that. So really interesting to see. And I just made this cartoon, like drawing. So this is just looking down at a caterpillar, and it's just the left side of its head. So those are just mandibles on its left side. And there's the brain in a caterpillar, and it's got an optic nerve because it can see through its larval eyes. And then it just shows that yellow is the epidermis, and the adult eye is going to form from the epidermis, and it folds it in like that, and it starts patterning the eye, and then when the animal pupates, it pushes that out and makes it a bulbous thing, and it's still patterning the eye. And then there's that later tissue maturation step that requires high levels of hormone. So what's going on is early on, there's an onset of that growth, and that growth is continuously requiring this low threshold, basal threshold, tonic threshold of either E or 20E. It's growing, growing, growing. It does all of this, that's differentiation, it's getting all wired up, and then the high levels of only 20E cause this tissue maturation like that. So just a really amazing thing like that. So anyway, we'll stop there. It's another example, but it fits into, you know, in a real personal way, into our table, right, of insect examples. And where it fits in is diapause, this overwintering plasticity. And that diapause uses a cue, like Juliet showed us, photoperiod, day length. And the caterpillars are monitoring day length, and they make a predicted adaptive response. And they've evolved that, like we've talked some about, and it was pretty easy to evolve because that basal level was running the whole show of metamorphosis. And if you stop the hormone, you stop metamorphosis. And if you bring the hormone back, it brings it back like that. So just really a cool example that fits into that whole table and other examples like that. Okay, so we'll stop here, and oh, we better check. And the recording's still going, so that's good news. And yeah, so on we go. So again, you know, we'll